Hello everyone, this is a lecture on autonomic nervous system, which is a part of pharmacology, systemic pharmacology. So let's begin. Okay, so autonomic nervous system, as we all know, is been divided into two parts: the parasympathetic nervous system and the sympathetic nervous system. The parasympathetic nervous system has the brain stems or the cranial nerves from 3rd, 7th, 9th and 10th, and the sacral segment from S2 to S4 spinal segments. Whereas in the sympathetic nervous system, it includes the thoracic segment from T1 to T12 of the spinal nerves and the lumbar segment of L1 spinal nerves. In parasympathetic nervous system, the ganglion are located closer to the organ, whereas in the sympathetic nervous system, the ganglions are located closer to the spinal cord, as they form the sympathetic chain, the sympathetic trunk. Next up, in the parasympathetic nervous system, the preganglionic receptors are nicotinic receptors, and the preganglionic neurotransmitters are acetylcholine. In the preganglionic receptors of the sympathetic nervous system are the nicotinic, and neurotransmitter is the acetylcholine. It is universal. In preganglionic, you will only see the nicotinic receptors and the neurotransmitter as the acetylcholine. Now moving on to the postganglionic receptors and the neurotransmitter. In parasympathetic nervous system, the postganglionic receptor can be muscarinic or nicotinic, and the neurotransmitter will be acetylcholine. Whereas in sympathetic nervous system, the receptors of the postganglionic neuron can be alpha or beta, where we have further subtypes in both of these we will learn later on, and the neurotransmitter as norepinephrine. Now moving on to the parasympathetic system, and the first thing, that is the neurotransmitters we are dealing with. So in parasympathetic nervous system, it is universal. The acetylcholine is the neurotransmitter, only neurotransmitter, and which is present in both pre and postganglionic neurons. So how acetylcholine is being synthesized? So in the preganglionic nerve ending, inside the mitochondria, the acetyl coenzyme A combines with choline, which is being uptaken from the systemic circulation. And in presence of choline acetyl transferases enzyme, the acetylcholine has been synthesized. In presence of choline acetyl transferases, acetylcholine has been synthesized. This acetylcholine is being transported and stored inside the vesicles. So when the calcium enters to the calcium channels, there is depolarization in the membrane, and thus leading to the opening of the vesicles or the ruptures of the vesicle, leading to the release of acetylcholine. This acetylcholine released in the synapse are being uptaken or acted on the receptors present in the postganglionic neuron, the muscarinic receptor or the nicotinic receptors. So this is how the cycle goes on and the impulse is transmitted. Whereas the other things that should happen, we have learned the synthesis of acetylcholine. So moving on to the metabolism or the degradation of acetylcholine, the excessive acetylcholine. We have a certain amount of impulse should be generated, and after that the excessive acetylcholine should be degraded. So for that we have another mechanism, which is the metabolism of acetylcholine, which for that we need an enzyme known as acetylcholine esterase. It looks like this. It has two sites, and the esteric site and the anionic site. The esteric site actually attracts the acetyl part of the acetylcholine, and the anionic site attracts the choline part of the acetylcholine, thus leading to the breakage of the bond, and thus acetyl is being attached to the esteric site. Whereas the choline is being released in the circulation, and thus been uptaken by the choline receptors back to the systemic circulation, again released, and the cycle goes on like this. So this is the story of the neurotransmitter in the parasympathetic nervous system. Now moving on to the receptors in the parasympathetic nervous system. This chapter is quite a lot interesting, and that much amount of hard to to remember, but if you understand it nicely, you will get it very easily. So the receptors in parasympathetic nervous system, we know there are two kinds of receptor: the muscarinic receptor and the nicotinic receptors. We denote it as M and N. In muscarinic receptors, these are G coupled protein receptors. I'll make a video on this as well, the types of receptors present in our body. So it is a type of G protein coupled receptor, whereas the nicotinic receptor is ion gated channel receptors, especially the sodium ion gated channel receptor, the ligand channel receptor types. So I will deal with it later on. So the nicotinic receptors, first of all, these are further have two subtypes: the muscular type and the neuronal type. It has been denoted as NM, and the neuronal type is been denoted as NN. Now, the location where the muscular and the neuronal are present. The muscular subtype of nicotinic receptor is present in neuromuscular junctions. And it helps in muscular contraction. As we know, it is ion-gated channel, so it will help in contraction. Positive effect. The stimulation will occur. Now, the neuronal type of nicotinic receptor, the location, it is present in three different locations. First, that is adrenal gland. It helps in increased release of epinephrine in ganglions, post and preganglion, as we know. So it helps in conduction and regeneration of action potential. And in the CNS, it helps in excitation, mainly the cognition of the brain. Now, moving on to the muscarinic receptors, there are five subtypes of the muscarinic receptors: the M1, M2, M3, M4, M5. Why I have written this in this way, all the odds one side and all the even this side, is to explain you their subtypes, their type. So the odd ones, the M1, M3 and M5, are GQ subtype. Again, these are G protein couple receptors. So the G stands here for G protein couple receptor, and further we know that G protein couple receptors has a further subtypes. The Q is for stimulatory types. The receptors which are M1, M3 and M5, this will no doubt stimulate any function, any part of the body where they are located. Whereas the M4 and M2, the even ones, these are GI type. GI, the inhibitory types, the G protein couple receptors which are inhibitory type. So they will, wherever they are, they will decrease the effect or inhibit the effect. So first of all, the M1 is present in two locations, the CNS and the GIT, the gastrointestinal tract. So in CNS, central nervous system, it increases the calcium concentration, leading to the increased production of action potential, whereas leading to the increased cognition of the brain. It helps in cognition of the brain. Now, in the GIT, it increases the secretion. Again, I've told you it's GQ subtype, so it will all do the positive effect. It will stimulate, increase the secretion and all, right? So it will increase the secretion of the parietal cells mainly for the release of HCl. Now, the M3 type. The M3 is located in various organs, so just remember this. This is important, very, very important. First, the GIT. It helps in contraction of the muscles, increase the peristalsis and increase the secretion in the GIT. Now, in bronchi, in bronchioles, in the lungs, it helps in bronchoconstriction. In bladder, in urinary bladder, it increases the bladder contraction and contractility, and thus lead to the micturition reflex. The next one, it is located in the circular muscles of iris. Now, this is a nice mechanism you have to understand. In the eye, in the iris, we have two muscles, the circular muscles and the radial muscles. The circular muscles helps in contraction, and the radial muscles helps in dilatation of the pupil. So, when there is stimulation of circular muscles, there will be contraction of the pupil. So, it will lead to meiosis. Meiosis is for, there are two functions they occur, the mydriasis and the meiosis. So, in this, it will be meiosis, and the radial muscle causes the mydriasis. We'll see later on. Now, The blood vessels, in the blood vessels, the M3 receptors, they are present in two things, the epithelial endothelial cells and the smooth muscles. In endothelial cells it leads to the vasodilatation, and in the smooth muscles it causes vasoconstriction, but the prominent effect is of vasodilatation as compared to vasoconstriction, remember this. So overall in blood vessels it causes vasodilation. Now, the M5 type is present in CNS, and the M4 is again present in the CNS. Their function and their function are still unknown. Now, the M2 type, this is very important. M2 is present in heart. Just remember, the heart was not actually, the M3 receptors weren't present in the heart, but the M2 are present in the heart. So remember this, and they help in inhibition of the ANS of the heart. ANS, the autonomic nervous system present in the heart, that supplies to the SA node or the myocardium. Now, the M2 receptors are more likely to be present in the SA node, so they supply the impulse more to the SA node and causes more inhibition in the SA node as compared to the myocardium of the heart. So what it will do, SA node and the other nodal bundles, so it causes blockage. Now why it's causing blockage? Because it is a type of GI subtype receptor, so it will cause blockage. When it causes blockage of SA node, it leads to decrease in heart rate, and when it causes blockage in the AV node, it leads to decrease in conduction velocity and increase in conduction time. Just remember this, and it's easy to remember. Okay, that's all for today. Thank you.